Hello learners, myself Dr. Renuka Mainde, corporate nutritionist and counselor in IGNU for their MSc Dietetics and Food Service Management course. Today we are going to learn about catechins and tannins. They belong to polyphenol group. Polyphenolic compounds from the natural sources such as foods and edible plant have been widely studied because of their potential health promoting and curative properties. Of the naturally occurring phenolic compounds, catechin and their polymer procyanidins are found mostly in green tea and red wine. Catechins is a member of flavone group of polyphenols, are best known biologically active component of green tea. Green tea catechins are the strong antioxidant. Similarly, now tannins, which is commonly referred to as tannic acid, is a water soluble polyphenol that are present in plant food. The major source of tannin in the diet include tea and betel nuts. Phenolic acid, hydrosoluble tannins and flavonoids have shown the anti-cancer, anti-mutagenic effect since they act as a protective against DNA and also against the free radicals by inactivating carcinogens, inhibiting enzymes involved in pro-carcinogen activation, and by activating of symbiotic detoxification of enzymes. This particular modules will be learning about tannins and catechins both, its structure, bioavailability, physiological metabolic effect on health. Let's now begin with tannin first. Tannin is an astringent bitter plant polyphenolic compound that binds to and precipitates protein and various other organic compounds including amino acids and alkaloids. This module highlights the structure, classification and properties of tannins. Tannins, commonly referred as tannic acid, are water soluble polyphenols that are present in many plants. They have been reported to be responsible for decrease in feed intake, growth rate, feed efficiency and protein digestibility in experimental animals. Therefore, food rich in tannins are considered to be low nutritional value. However, a recent findings that has a major effect on tannins was due to their inhibition on food consumption or digestion, but rather the decreased efficiency is converting the absorbed nutrients to new body substance. Let us now first understand about the tannins. Tannins, the term tannins refers to use of wood tannins from the oak in tannin animal hides in leather, hence the word tan and the tannin for the treatment of leather. However, the term tannin by the extension is widely applied to a large polyphenolic compound containing sufficient hydroxy and other suitable groups such as carboxyl to form a strong complex with various macromolecules. Tannins are the polyphenolic compound composed of very diverse group of oligomers and polymers found in plant parts including leaves, roots and fruits. They precipitate protein and also complex with the starch cellulose and minerals. Tannins have molecular weight ranging from 500 to over 3000. Tannins are found as shapeless, yellowish or light brown masses like powder, flakes or sponge. The percentage of tannins present in the plant, however, varies. The tannins are usually found in large quantity in bark of the tree where they act as a barrier for microorganisms and protect the tree. Tannins have ability to precipitate the solution of gelatin, alkaloids, glycosides, heavy metals and proteins. Because of accumulation of OH group on small size nucleus, they have antioxidant nature. Tannins have a property to react with mucous membrane and cause precipitation. Let us now understand the chemical structure of tannins. These properties of tannins are based on their chemical structure having 2 or 3 phenolic hydroxyl group on phenyl ring in the molecule of moderate size. Tannins have many these phenol groups as a part of their structure which can react. The chemical linkages of tannins to proteins are interesting. Tannin protein interaction are influenced by characteristics of the size of protein, that is the size of amino acid composition, etc. The characteristics of tannins, whether it is size and structure, the condition of reaction that depends on pH, temperature, solvent and time. The water molecules form a hydrogen bonding with protein in their places. As the tannins have large number of phenols, they bind with many proteins at different places, thus locking the proteins together. Some tannins are closely associated with D-glucose, the sugar. Now let us understand the types of tannins. First we will see about hydrolyzable tannin. This is an ester of sugar and phenolic acid or their derivatives. The sugar is usually glucose, but in some cases polysaccharides have also been identified. Acidic, basic or enzymatic hydrolysis often occurs spontaneously during extraction or purification. At the center of hydrolyzable tannin molecules, there is a carbohydrate, that is usually a D-glucose. This type or a tannin is further subdivided into allagitannins. Gallotannin. Examples of gallotannins are gallic acid, esters of glucose in tannic acid, and their structure is C76, H52 and O46, found in leaves and bark of many plant species. Second type of tannin is condensed tannin. This tannin is certainly more important than hydrolyzable tannin. Much less is known about their structure and many aspects have yet to be identified. This type of tannin products are tannin reds while boiling with acid. Traditionally, most commercial source of this type are heartwood of cubracho, bark of wattle. This has been used in the leather process industry to get better type of quality of leather. At the same time, various biological and pharmacological activities related to health effects of tannin with the variety of chemical structure, including those of smaller molecular size, have been found. As for the molecular size, epigallocatechin gallate and epicatechin gallate, the main tannin in the green tea, are the example that exhibits the property of tannins in spite of their small molecular size. They exhibit binding activities towards the protein and other substance and appreciable antioxidant activities besides their anti-tumor effect. Now, condensed tannins or Pro Anthocyanins are the polyhydroxyflavanol oligomers or the polymers. They consist of flavon 3-OL or flavon 3,4-diol monomers that are usually linked by carbon-carbon bonds in 4:6 or 4:8 position. Type B Pro Anthocyanins are classified according to hydroxylation pattern, chain of extender units, and most common in the plant foods are procyanidin, prodelphinidins and proplargenidins. The name Pro Anthocyanins is derived from the fact that when treated with acid, they react to form insoluble flobefins and red color anthocyanidin solution. Type A tannins and related polyphenols of constant structure and composition are all ellagitannins exemplified by crystalline monomeric gyranine and dimeric argimonin are type A tannins. Now type A tannins also include epigallocatechin gallate, epicatechin gallate, which are easily isoable main components of green tea tannins, are exceptional members of polyhydroxyflavone family exhibiting tannins activity despite their small stable structure molecules. Type B tannins, which have variable mixture of analogous polyphenols. The structure and composition of this type of tannins obtainable from particular species of plant and are not always same, as their structure and composition differ seasonally and also depending on the growth condition of the plant and on the methods used for the preparation. Let us now conclude, learners. Hope you have understood the complex structure and chemistry of tannins, which belongs to a large family of polyphenols. Extensive chemical, biological and pharmacological investigation of tannins, particularly those of ellagitannins and their congeners and also related to polyphenol of various constant structure, are changing the concept and significance of tannins in human health. These discoveries are opening a new way to classify and study the chemical, biological and pharmacological properties of tannins.